Welcome to Focal Points. Today, our special guest is Dr. Kevin McCairn. He has found some deeply disturbing findings. In fact, he has identified amyloid fibrils in a 3-year-old who was exposed to the Pfizer mRNA injection in utero. He was born without vital signs, and he has been chronically ill since. And now, at 3 years old, it does appear he has these amyloids in his blood. This raises all sorts of implications, and Dr. McCairn is the one that identified it, and he has done a lot of work, so much work, in really identifying what this novel pathology is. And so, Dr. McCairn, give us a little brief background about yourself, and let's dive into these novel findings.
Sure. So I'm a systems neuroscientist, 25 years under my belt now. I specialized in corticobasal ganglia disorders. I developed the first primate model of Tourette's, geared towards treatment of young children, but that work wasn't funded very well, so I had to do a lot of other work around neurodegeneration. So I would build primate models in that domain, Parkinsonism in particular, but they have overlap with the disease type which we're looking at here. Neuroscience started to converge after the sort of 2010 era, that there was overlap between the canonical neurodegenerative disorders, so Alzheimer's, Parkinson's, and what most people think of as the scary end of the neurodegenerative spectrum, which is Creutzfeldt-Jakob disease. And basically all that is not all, but in the disease process, proteins will oligomerize, misfold, and basically build inclusions, intracellular or extracellular inclusions in the central nervous system. Now, amyloidogenic disorders are not just restricted to the above the neck. You get them elsewhere. And case in point, I want to say it was it Simpsons creator just died of cardiac amyloidosis this week.
Yes, somebody did. Some notable figure did.
And so the concern has been not only with vaccines, but this started with the virus. I was very early out the blocks. One, just because of my early contact with the virus itself, and obviously the storm that blew up around that, which was this was a synthetic chimera that was, now we know, is the consequence of biowarfare research, heavily funded by DoD, all those DITRA, DARPA organizations. They've been funding this to the tune of billions of dollars, and somehow they were funneling money to the Chinese as well. And so it generally just I view this problem as one of whether people were hurt by the virus or the vaccine. It's a consequence of exposure to biowarfare agents down either side. That's how I sort of view this. And in terms of, moral harms done to people, in terms of the vaccine, you could, at least there was this sort of, you could say the state mandated people's exposure to these agents, which were not novel vaccines in the way people understood them. They're based on gene engineering technologies. And the fallout from that, I think, will be picking apart for decades. And yeah, beautiful slide, not mine, but yeah, particularly along the bottom there, I hold a heavy grudge against what they have done. And this all sprung from 9/11. We're getting off track here, but this all sprung from the anthrax attacks, 9/11. That was American biowarfare agents out of Fort Detrick, and they built out they took the brain trust and pumped out billions of dollars into the universities to look at this tech, and now we're dealing with the consequences of it. And so probably the best thing to do is just jump into the slides, and then
Yes.
I will.
Yes. So this biopharmaceutical complex and the government biodefense sector has committed which appears to be serious acts against the population. So let's
against
see.
humanity.
Yeah,
that's where they sit in my view. Will we see any justice? I don't think so. I think the forces that are arrayed against the people are probably a bit too much and a bit too slippery, and they'll change their corporate face again and again and again. You'll be chasing them for decades, I'm afraid. That's the unfortunate part of it. But we can do something about trying to understand the impact that it's had. And so what I do, and what I've, like I say, I've been 25 years working with, well, it's basically central nervous manifestations of amyloids for Parkinson's. And now I've been pulled into how that they're expressing in the population at large. Yes, there's neurological impact that's linked to vaccines. Very large population study that came out of Korea last year said that there's significant cognitive decline. What else did they list? Alzheimer's, of course, but these are very large numbers, and statistics quite valid. They have very good record keeping there. I know I live there. So yeah, on the screen there is just my lab. I work with animals as well. We're trying to model in hamsters, white hamsters in particular. Their ACE2 receptor is similar to that of humans. Do I have control of the slide? Does that let me do that? Okay, so I'll just do that. So what I'm going to do is I'm going to start and I'll tell a story about how we got to subject B3, baby B, I like to call him. And in the process of working around these animal models and looking for therapeutics, etc., I came into contact with Richard Hirschman, and they said, would you, because I was seeing these come up in sort of Twitter feeds and what have you, and I was saying, if that looks very amyloid-like, please send it to me and I'll make sure that it gets processed properly so we can at least get some better answers. And so what you can see on the screen there, I got a bunch of samples, some in formalin, some stored under argon. The more pink looking one is the one under argon, and basically looking, so we'll start just going from the macro down to the micro, nanoscale, and then we'll look at the images that we see from baby B. Okay, so it doesn't scroll with wheel, that's annoying, but there you go. That's me just taking one of those clot-like substances out, just pinned it under the scope with 30 gauge needles, and then oh yes, of course, I put this in because I've been a bunch of substacks that I've done, but I've been working with Kevin McCairn as well. We wanted to confirm that that was human tissue. And that's why this slide is here, and that confirmed that we're looking at human tissue because it had the genomic signature for human RNA, RNA-dependent polymerase, you can basically think of it as. And that sort of says this isn't something they've pulled out of an abattoir or something. It's coming off. There's a genomic signature for human tissue. Okay,
so it's human, right? It's human.
Yes, yeah. There was slight signals for vaccine markers, but it's weak, and so we're not leaning too much on it. Kevin is like, he's not confident in that data right now at the moment, but there's more to come in that space as we check more and more of the samples. And this is us now zooming in and looking at the clot through a dissecting scope. Now we can see it looks less smooth. There are ridges and sulci and grooves in that structure, which kind of tells you that it's, hasn't just been pushed through like a Play-Doh type sausage maker or something, right, to just squirt out some protein. That seems consistent with coming from the inside of a blood vessel, which is what Richard Hirschman claims. This is me taking a scalpel and just cutting through the section, and basically people have complained about this image because of the lighting, etc. It looks a little different, but that's basically the flat surface through the clot. There's no lumen. This isn't the blood vessel itself. So typically clots tend to be I'm going to say periluminous, right? So if you have an atherosclerotic plaque, it grows in the lining of the blood vessel. This is not. This seems to be filling and packing the blood vessel itself. And that's basically why you're seeing the sort of contours that you are slightly.
It looks like a piece of salmon.
Yes. If I had, when I describe it to people, I would say just think a little bit like fake crab meat. but not able to sort of that sort of consistency. But imagine not being able to sort of peel little strips, right? But in terms of feel, and it's like that. So in order to sort of investigate this further, you obviously have to process it properly, and to do that you need something called a microtome. In this case we're using a freezing microtome, and there you can see, so that's the device, sort of washing machine size, the inside of the device, and there are slices that I've cut, 3 slices. We'll be taking a closer look at those. And this is at the lowest power of magnification. So what looked like fairly solid tissue when you were looking at the macro scale, as soon as you get it onto the microscope, where all those white patches are is basically gaps. And as we go up in magnification, you can basically see it's a very sort of fibril-like appearance. I've sort of likened it to mycelium, if you've ever looked at it under a microscope, but it's not mycelium, it's fibrin, which is one of the clotting proteins that everyone has. You need it, and there's lots of it in the blood. So this is, it's very, you're already learning a lot just looking through a light microscope if the sections are prepared properly, but there are tests that we can do to say what type of protein are we looking at, and we're interested in, is the protein a misfolding amyloidogenic protein? And the way that we do that is we use selective dyes, fluorescent ones in this case. It's called fireflavin. And oh yeah, just I'm just getting ahead of myself, but this material, if you just put it under UV light, autofluoresces. And so work by Pistorius and Kell have identified microclots. They're much smaller in form, the sort of hundreds of micrometer range, but they've, in long COVID, also in vaccine injured, they've got a manuscript out recently, but they process their material such that they see the very, very small forms, and it's centrifuged, and they're looking at plasma, whereas this has come from a direct aggregate in the blood vessel. And we get the same autofluorescence. So that green coloring that you're seeing there is it being hit with UV light with a blue filter, and it glows green. And then on the left hand side we've added fireflavin, and what you can see is there's like a polka dot appearance to it, and you can also see a sort of fiber like structure down here that has a little torsional twist to it. I'd like you to keep these little fibers in mind as we sort of step through the size of the magnification, I'd say, rather than the size.
So because it's illuminating, what does that exactly tell us?
So first principles would say that when the protein is oligomerizing, so the protein is malformed such that hydrophobic beta-pleated sheets, right? So if you ever look at protein diagrams, you'll see the sort of sheet, they sort of run backwards and forwards, or you'll get sort of curly looking structures, and they're called alpha helices. And the protein tends to take that form up dependent on, it's a thermodynamic thing, and it's trying, and protein folding is just one of those areas. It's hyper complex. Why does a protein fold in its particular manner, etc.? And they put a lot of computing power trying to answer that. But in the disease state, something causes the protein to misfold. It has this core which doesn't like the aqueous environment, and then it bumps into other proteins of the same species, causes them to misshape, and one gets to cover its core, and then the other one has its exposed, and it basically a bit like a domino effect. And you're just looking at higher magnification here, and basically what you can see is, you can see sort of like the punctate areas, but the areas between the concentration as well is glowing as well. So there's sort of a fibrous amyloidogenic mat between all these different punctate inclusions. So the fact that we see fireflavin reactivity here says to me immediately, oh, that's sort of past one of the first tests that we would use to say, are we seeing a diseased form of the protein? Okay. So after this, we're able to go even further in terms of magnification, in this instance using scanning electron microscopy. And here we've cut the sections thinner, and section is sort of filling the bottom half. As you can see, these sort of folded sheets, etc. And we're basically going to zoom in. And the scale that we'll be looking at is like one fiber of that very fibrous mat that we were looking at the highest magnification in the light section, if that makes sense. And the scale bar is on the bottom here, right? So this is us going to 250 times. So we're sort of advancing to the limits of anything you'd get with a good light microscope or beyond. At 1,000 times, we start to see this fibrous pattern begin to emerge again. And, you know, it's a case of biology being sort of as above, so below, sort of fractal type appearance. And if we go to the next, which is at 5,000 times, we really get a close look at the peptide structures that are forming up this what's a complex hyperstructure in this clot of punctate regions of concentration and sort of mats out between them. And now we're looking right down at the substrate of the fiber, the building blocks of the fiber. And the important note to take from this image is that you can see that there's a twisting going on in the fiber. And that twisting is indicative, if you've looked at enough proteins under a microscope, etc., it kind of tells you if there's something wrong with its native form. And also these nodules that you see everywhere are abnormal for fibrin. And we'll just see normal fibrin in a second. But at this magnification, every level that I've seen says to me that that sample that has come out of the cadaver provided by Hirschman has all the characteristics of what I would expect to see with an amyloidogenic protein. The difference being is just the scale of them is far, far larger than anything that you would see, or I would have seen, looking for amyloids in a monkey brain, which are intracellular inclusion, so very, very small. These are, you're looking very small at this stage, but they're growing into superstructures, okay? And that's I'm presuming because of the nature of fibrin, which is to make a sort of macro molecule. But in this instance, it's growing and growing. It's malformed in its protein landscape so that it can't be broken down by the normal enzymes that the body relies on to disperse clots, right? So you have clots forming all the time and they're broken down all the time. And in this instance, they're going into this disease misfolded state, seeming to hang around, and I would say it's likely to be bad for your health should you be getting these aggregations. So I encourage everyone to go, and this came out last year, but it's a very important paper, Fibrin Drives Thromboinflammation in Neuropathology in COVID-19. And what do they show? So in this bottom corner, the left-hand corner, you can see normal fibrin, which basically looks like noodles, smooth noodles, sort of overlaying each other. And in this instance, they've exposed it to spike protein. What do we see? We see a twisting form and also nodules appearing on the protein. Okay, so there's, you know, we're starting to see some overlap, not just between what we're seeing, but what's published data and what sits within the framework of our understanding of misfolded protein, amyloids, etc. So if you have questions, just feel free.
So it's clear that the spike protein is doing this, right? At this point in time, based on prior evidence and now your evidence, it clearly seems that the spike is indeed causing this misfolding.
Yes.
Is that right?
Yeah, and it's not a contentious statement to make this day and age that the spike protein is a amyloidogenic seed or catalyst in this instance. Only where it seems to be manifesting, or the lowest hanging fruit with respect to its manifestation seems to be in the blood. In the case of in the vaccinated, in this instance, basically you've had a transfection get into the bloodstream, and once this misfolding cascade starts, a lot of times the thinking is it's hard to get rid of. And but we see the same thing with COVID, long COVID. I see very similar data as well. but it's spike, right? It's spike across the different conditions. One bypasses immune barriers, the other is shot right past it, and given you're sort of having a massive dose dropped in, and we see in some patients that they're producing spike years later. There was a Yale study out showing
So do you think the vaccine is contributing, would be a higher risk factor for this compared to the infection, or would you say they're equal? What would your opinion be?
I would say it depends on the individual. But if it was the choice for me, I would take the risk with natural infection and saying, okay, if I know that thing's out there, and try to take steps to mitigate this. Something synthetic that's the product of biowarfare research just shot straight into you through a new means of delivery. I would be inclined to wait on that. And this was something that was forced onto pregnant mothers, and you literally had, I want to say she was the ex-director of FDA, who basically was pregnant just a couple of weeks ago, saying, "Oh, I didn't take it because I didn't know what the effect would be on the baby," right? So that was literally the head. You had these people forcing and heckling mothers to go get these shots, and literally the people responsible are dodging their responsibility when they should have been front and center if they were so confident in this technology, and they weren't, right?
Right.
So
Exactly.
how are we doing for time?
We're doing great on time. Let's get into now what's really disturbing is the
Yeah.
consequence.
Yeah, so there's a couple more slides just about those clots, which is I've done elemental analysis. I don't see anything that would indicate heavy metals or anything. This just looks like abnormal protein folding. That's the quantification of that. Not really that interesting. But here, this busy slide is of interest. This is just the screen grab from Raman spectroscopy, which is looking at these samples. And there's a Raman spectroscopy basically gives you a signal around the molecular formation that you see. And there are peaks in spectral ranges that are associated with amyloid-like structures. And typically they're around 1680 centimeters. Ours developed a peak at 1720 centimeters. I can think of lots of reasons why that could be. The fact that there's a peak that close is suspicious in my mind. Again, there's lots of work that needs to be done in this area, but the signals are close enough. I've got all the other data that says to me that it's amyloidogenic, and I would say there could be molecules at the ends of the hairpins, the warping of the oligomerization normally that would give you the core, and as their biowarfare agents. When I'm sort of sitting there pondering this, you would do that because you would want to warp the landscape as much as possible, so it's harder for the body to break it down. Okay, so just keep that in mind. And so now we get to baby B, and so it's possible, if people would like to, it's capable. You can get a measure of amyloid microclot burden in the blood. They're well validated methods in terms of the UV dyes, and with my eyes looking over a scope, I can find these aggregations and give you a estimate as to what I think is a low, moderate to high amyloidogenic burden in your blood sample. Okay, so this sample was sent to me. This is just one of many that was in his blood, and it sort of caught my eye straight away. And one of the things that I'm always on the lookout for is a fibrillar twisting form, and if I see those, that nudges you into the high category, right, where I would say probably you want to be thinking about more mitigation methods, perhaps.
Well, Kevin, before we dive deeper into what they found in this kid, let me just quickly upload this and just describe to us what exactly happened in this case. Now, I made that figure up top there, right? So that's just to just so we clarify to the audience in case, you don't
Mm.
approve of that figure, but
That's great.
so tell us exactly what happened to
Yes.
this kid.
Yeah, so he was in utero. The mother, 32 and 34 weeks, no, it's 35 weeks that he was born premature, no vital signs. And it was 2 shots of Pfizer. And yeah, they've been concerned about the child since. And they saw me talking on Steve Kirsch's stream, and sent me a sample. The instructions are really easy. It's literally just like a blood test for like a diabetic would do, a couple of spots of blood on a glass slide, and I can usually give you a indicator as to what's going on. And so yeah, first of all, you're looking at the light microscopy on the bottom left hand corner. Then next to that is the UV dye being added, and then looking at it with scanning electron microscopy just to look at the fibrilliform. And in this instance, are we taking a leap by saying this is definitively amyloidogenic fibrin? In this instance, I would say that there's enough published evidence about that form of pathology that there is consistency in the methods and validity to those methods, that in this instance, if we see even one safety signal in a child's blood, it's imperative that this discussion goes out as far and wide as possible. The fact that this hasn't been, please dismiss it, right? Do the work and dismiss that this is a problem in people exposed to these agents. Again, for me, it comes from both sides of the aisle, whether it's their chimeric demons that they're breeding up in these labs, to the synthetic version that they're sticking in people en masse. And it's 13 billion doses into 5 billion arms. Even 1% of that is a massive number in terms of health impacts and what it could do. And
So we know this child was exposed to the mRNA genetic countermeasure in utero, right? We know it because we know it transfers through the
it does
placenta.
get past the placenta. It does, yeah. All that work's been done. You can find the citations for that. I put that in the Substack. I didn't overload the Substack like I would if I was writing a manuscript for academic journal. It's there just to sort of guide people. Hey, look, this is what we're seeing. There's already prior evidence that this could be a problem. And what do we see when we look in a child that's suspected of receiving an assault in utero? Well, I'm seeing these amyloidogenic fibril that concord with a microclotting phenotype.
So should this not be in a 3-year-old, like, right?
No,
Can this happen normally at all? Like, can this
no.
happen not from SARS-CoV-2 or the
Well,
injection?
you can't say never, but there are cases of like disseminated intravascular coagulopathy, like sepsis, etc. When things like this happen, maybe the child had that. But right at this instance, if you were to do the investigation and ignore the precipitating events and just say this is something that's happened as a consequence of exposure in the environment, okay, you're not doing your job properly. Do I accept that there could be environmental factors that have done this? Maybe one of those infections they had to treat him for tonsillectomy or middle ear did it, potentially. But you know what? First off, let's just rule out that it wasn't a man-made intervention forced onto pregnant women over the last 5 years. Like, can we at least agree that we should do our due diligence in that area?
Right. I mean, and he was born lifeless, right? This
Yeah.
child was born without any vital signs, so he was probably damaged clearly from the genetic injection. And then he's been chronically ill since. He had multiple surgeries, right? He's had
Tonsillectomy, middle ear surgeries multiple times, doesn't recover well from seasonal viruses, seems to have basically a sort of immune dysregulation. And he's growing well. The developmental markers seem to have been hit, but what's the long-term outcome for this individual when already there's this disease burden placed on him that perhaps wouldn't have been there without the intervention in the first place.
So what are the implications of this? So yeah, so that it does appear to be an amyloidogenic fibril. What are the implications that that was detected in his blood for not only him possibly, but for all the other mothers that were inoculated with
Well,
the bio warfare
it's
injection?
population wide in terms of what are the consequences to amyloid exposure, environmental biowarfare, amyloidogenic peptides being seeded into the environment. In this instance, where they're leaving a footprint in blood and causing dysregulation in clotting. All manner of disorders can begin to stem from that. And if we looked over the normal trajectory of someone's lifetime, you would say someone who has this type of disorder is going to be prone to degenerative conditions, cancers, cardiovascular, neurological. The list goes on. And the problem with it is because of the it's obfuscated exactly how the disease will present because of the systemic type exposure that's occurred. And generally, you can think of just the weak points that a person may have. And it just may be another infection that he gets at some point down the road that just puts him into that chronic ME/CFS like post-COVID state. And we don't know. Right? This is the thing. We don't know. There's not good evidence with respect to their safety. Well, no, I'll rephrase that. The argument from the authorities is that we've done billions of shots. There's no problem. Is that really true? Have we done the checking? There's no checking for this type of pathology when they do the normal tests on, a child as is born, as they're developing, etc. So this would be missed. Just, oh, he died at birth. Maybe they'll say, well, maybe it was a vaccine, maybe it wasn't. Well, I'm not prepared to just let that slide in this instance where I see slide after slide of people's blood who have been vaccine injured, confirmed vaccine injured, right? The diagnosis from doctors, etc., or people that have long COVID. Now, this is an extreme example that you're looking at here. Why his blood really formed these very, very precise fibrils and microclots clustering together? I don't know. Again, you would need far more investigation, etc. But again, this is preliminary investigation.
This is a sentinel detection, right? This is the very first detection, specifically in someone so young, right? Nobody's done it, but you're the first one to do it. And now people really do need to investigate. Particularly, I would really hope that our public health agencies would take this seriously. But looking at what they're doing now, it doesn't look like they will.
No. What their goal is to get each person, they'll gaslight each person that turns up ill, and they'll get banded around the health system, and they'll scratch their chins and, oh, it might be this, might be that. And primarily, I think what you'll see is this sort of long COVID phenotype develop, right? Exhaustion, brain fog. And that can be from as simple as just the microcirculation being blocked up with these microclots that just the body has a job to get rid of. And if the macro clots that we were looking at earlier are any indication, then some people are building huge reservoirs of them.
So is this possible that these small little fibrils can start to kind of aggregate and bundle up into those large white clots?
I think so. I think that it's one hypothesis that needs to be rejected right now. For the moment, I would say there's all the indicators are that it can and will.
Does that mean it's what's called a prion? Like, do you think that this is a prion type pathology at this point?
All prions are amyloids. Not all amyloids are prions. That's my saying. I've heard people stealing it from me, but I pulled that one out. But all that means is prion means proteinaceous infectious particle, and the core pathophysiology in this instance, when they talk about prion, Creutzfeldt-Jakob, chronic wasting disease, they're basically talking about misfolding occurring in the prion protein. In this instance, it's an amyloidogenic oligomerization, this time in fibrin, the clotting mechanism. The problem here is that you have so much fibrin, and so the scale is way different to what you see with when you're looking at brains that have been building. These are sort of membrane bound proteins, often the prion protein, and they build these sort of small difficult to digest aggregations, and you have to look under a microscope to see them, whereas this is nearly visible to the naked eye. And that's just a consequence of the environment and the medium in which it's occurring. And the thing with amyloid type conditions is that you have what's called cross seeding. So basically just think of what the misfolded peptide is coming into contact with. It could potentially just cause other proteins of other species to misfold as well. And often when we look at these aggregations, if you took alpha-synuclein 1s or Lewy bodies, etc., you'll find a mix of proteins in there. And it's the same here. It's not entirely fibrin, but it's the medium in which it's occurring in the blood. High volume, high, a lot of it's moving all the time, and so the chances of sort of sticking and aggregating together are going to be higher. And
so this appears to be something novel, right? So that we can't really call these prions, right? But we can call them prion-like.
Prion-like, yeah, they have the same mechanism. Amyloidogenic is the technical term. They're amyloidogenic forms of fibrin, right? And this has been described by, again, Pistorius and Kell. They were the first, and they should be recognized as sort of identifying this phenomena, not only with COVID, the virus, but now they've got a paper, I think it's still in preprint, but they're seeing the same with vaccine injured as well. And, the form that I've identified in is just a methodological difference between how we're looking at them, which is if you spin blood down and you're looking at just the plasma, you're basically doing a size exclusion anyway, and you're moving heavier stuff down to the bottom. So it's unlikely that you would see these big aggregates. You're likely to see the smaller forms and fibrillar forms as well, I've seen. And we're in the process of sort of cataloging them and trying to find better ways of detecting them and ways of sort of tracking patients across long time periods, and using the amyloid microclot as a biomarker for the health, the disposition of the patient. And
Well, since this was in his blood, right? That probably means it was circulating, right? And it could have deposited in various organ systems, right? So this could be in the brain, right? This could be in the heart. But the most concerning thing is we don't know how many kids that this is going to be detected in, because nobody's looking. Nobody's looking except what? Your team, and how many other teams? Are there other teams looking at this?
There's a team in Germany, Greece, Switzerland, the US. There's one, Dr. Vaughan.
All right, so we got some people looking, so that's some good news, right?
And we're all seeing the same thing. That's the thing. It's and of course Pistorius and Kell, they have institutes behind them looking at this. And is there a sort of immediate action that could be taken? And what seems to be the sort of the gold standard is you can think of it as like dialysis. Take the blood out, filter it, try to remove these larger aggregates, and put clean blood back in. But is that a permanent solution? I'm not so sure. And there's many other, and I'm working on different therapeutic avenues here. So flavonoids, sort of, you're trying to sort of stop people going down that sort of escalator into more and more worse symptoms, right? And so you want to make people aware that there's an issue out there, so they make the right lifestyle adjustments and then supplement that they can put into their normal regime. And then there are more and more medically orientated interventions you can make. And
So what do you think, like, just for say some natural compounds, what what could possibly be effective against these
So
amyloidogenic
I
fibrils?
can point you to case studies where, so what appears to happen in circulating blood is the primary blood monocytes aggregate spike protein, have a lot, and can carry a lot of the sludge that comes from infection, I guess, or transfection, depending on the case. And so that's Joachim Gerlach in Germany, medicinals. I've seen their case study work. There's the work from Pistorius and Kell, where they're doing, I think they're doing like the dialysis type approach, triple coag therapy. So Eliquis, maybe throwing some aspirin, nattokinase, that type of thing. I think it's a case of just try to do something till the patient reports, I feel better in this instance, and just again try to prevent that crash where they just get into this inflammatory loop, and it becomes very, very difficult to get them out of it. You know, one of the things I'm working on here is stem cell approaches. That seems like on the bench that works great. It's just not really, it's not scalable therapy. Yeah, if you want that, you need to come to Japan and get that treatment. And it's, you know, you don't want to be doing that to a child, from my perspective. So the thing to do is detect, and then you want to make sure that the child isn't getting into an inflammatory or environment in which they're picking up infections time after time. They're probably going to need a bit of shielding and protection in this instance. Yeah, it's an ongoing problem where there isn't a nailed on solution. And the first thing is you have to recognize that there's a problem. And, you know, the problem is I get pulled between it's a case of it's all vaccine or it's all virus, depending on which side of it. It's not, it's both. Right. And this is the worst I've seen. And this, I say, is pointing at the in utero exposure. But generally, on a case-by-case basis, it's very difficult for me to tell the difference looking under the microscope. If someone's ill and they have this
Right,
amyloidogenic.
right. Well, Kevin, I want to thank you so much for the very important work you're doing and coming on this show and sharing these findings with the public. It's extraordinarily important because you're doing the work that our public health agencies should be doing. And so, what are your final words to our audience? Where can they find you? Where can they follow you? And what's your take home message today?
First off, it's done. And so to keep calm would be the first thing. No need to. This child generally is not bedridden. There's just a pathology that we can detect and some immune dysregulation that we see. If you want to have a test, it's relatively easy. Go to my website, synaptechlabs.com, and there's a send blood samples. There's instructional video how to do it. You put a couple of microscope slides in a case like this. I don't know how well the spot sort of comes through. Put a couple of spots on, and it's enough for me to find a signal. If there's a signal, then I can tell you, and then you can think about the methods that you would use to try to make an intervention. With children, it's hard, right? Because you can't even get them to eat properly. So trying to get them to, you know, you got to take your turmeric, and that ain't happening with a 3-year-old. Wouldn't happen with mine anyway. I know that. So yeah, I think it's more sort of like lifestyle adjustments. Know that there's a problem. Track the problem is an important one, right? So if there is something there, is it just there at that time? Is it just it's post another infection and they're sort of a bit inflammatory? These are the things that should be being put into place, but they're not, right? And there's no motivation on behalf of the authorities to do this because by doing so, they admit that there's a problem. There was a problem with how it was rolled out. There was a problem with how they were conducting scientific research and spending tax dollars, the runaway bureaucracies of the defense sectors, etc. Those are huge forces, and none of them will position themselves in such a way that in any way they will receive any type of blowback. They'll kick the can down the road till baby B is 25 and he's going to the doctor and saying, I'm run down, I can't do anything, brain fog, etc. And then that's the situation I see. And you're not gonna get Pfizer into court beyond probably a few worst cases. It's grim in that respect, but we have the tools to make the detection, and that's a good thing in this instance. We're not entirely dependent on what these bureaucracies say in this instance.
Right,
So
right. Thankfully, we have independent scientists like you and like many others that are doing this important work, which is absolutely critical, absolutely vital, and our public health agencies, this biopharmaceutical complex that creates the pathogen, the synthetic pathogen in their biolabs, and the genetic countermeasure that it
mm,
also induces this damage. And
mm,
both of them are pushed onto the global population, and now it's resulted in this absolute disaster of now what appears to be this new amyloidogenic pathology that
mm,
we're just now uncovering
mm.
in
Well,
a
I
lot of
won't say
people.
just now. We've known from very early on, but it's been constrained by, well, just there's the pushback within academia. It's just an infection, etc. There isn't this broader view taken of just there's been a mass exposure, and that's it's just a sign of the corporatized oligarchy that we live in today. I don't know. If people aren't black pilled enough yet with what's going on, I mean, just look what's happening today, right? Let's shoot off on a tangent, but
Oh yeah,
you know,
we're black pilled. We're black pilled after the FDA approved next spike. Next
yeah.
means death in Latin.
Mm.
Yeah, we are truly black pilled at
And
this point.
that's the worst bit. That's the N-terminal domain, the S1 segment, right? Which has this really nasty 20 amino acid segment on it, which does catalyze the scrapey prion, funnily enough.
Oh
And
no, so no way. So next spike is actually worse than the full length spike.
yeah, I would say so. Yeah,
Oh
if when I'm
man, so
when I'm doing
that's why they use the Latin term for death. Then that makes absolute sense.
if I'm working on the bench, right? If I want to get reactions versus whole spike versus the S1 segment is far more. I'll get better data using that. And
Oh man.
yeah, no, when I saw it, I was just like, well, am I shocked? No. Just how can people just keep going back to the same well and drinking this slop? I'm sorry, it's like that's their problem now, right?
So it's true. This one truly is though. Like it is novel, right? So they only tested it for immunogenicity,
mm,
but it's a completely new mRNA that is encoding for the N-terminal domain,
mm.
and but that's never been tested in humans.
Well, I only in the sort of fragments that come from the transfection itself or the infection. And
Right.
it's that S1 segment that gets cleaved at that furin cleavage site, and generally it sort of springs off and goes into the cellular environment and causes toxicity. It wants to make an environment in which the virus itself can become permissive. That's why these very short reactive epitopes evolve. And just to fill your audience in, a lot of this work was done around HIV. Okay, and that's been fed through weapons development, and Los Alamos has a lot to do with this, and they're looking, and what they try to do is they try to shrink down and peptides that are known for a viral pathogen, and in this case HIV. They try to shrink down what is the minimum that you need to cause, and HIV is a highly amyloidogenic disease, right? In the end stages, you get HANS, which is HIV-associated neurological disorders, and the brain shrinks, and you can put those peptides in the brain, and they cause amyloid cascades to occur in the brain if you directly put them. And I think that they've spent billions honing this down, trying to understand this these reactive surfaces. And this is the consequence of biowarfare research run amok. And
So
what's
humanity
really
was attacked, clearly. I
yes,
mean, what? So yeah, they've been working on this tech for decades. You're right,
mm,
and then they finally honed it down, and now we're paying the consequences
mm.
of their
and
reckless actions.
how it works. Right, so this is a really dark thing, right? So the the response to these reactive epitopes, right, can basically be classed as work into incapacitation agents, because generally the response to them is not immediately lethal. It's this, you'll get the sort of initial disease state, and then you get this prolonged sort of recovery as the body's trying to adapt to the new environment that's been forced in on itself. From a military perspective, if it hasn't killed you, then wow, this is we've got something useful here, that if we can throw that onto a battlefield. And the idea is why they do this geographic genome-wide sequencing, is you're trying to find a pathogen that's endemic to an area. You tweak it so that your side has a countermeasure, and then you smother the area that you're going to go into battle with, in the hope that you knock out 30% of the fighting force. That's their thinking. And it's the honing of these epitopes and delivery of them is what they've been aiming for and using people's tax dollars for. It's gross.
It's disgusting. It's
Mm.
disgusting. Well, Kevin, thank you so much for coming on. Again, this was an absolutely fantastic show, vital information.
Mm.
And please keep us updated with your new findings, what you're looking at in the future. And so
Oh
what? So
yeah, I mean,
everybody
I've got
can
a whole
contact.
bunch you can look at. It's not just freaky looking aggregations in blood. There is the therapeutic interventions. We can look at that, animal models. The issue is just I'm independent, right? So the way that I survive is just people hit the tip jar basically, and I've managed to get by like that and speak publicly as a consequence, and I take that responsibility seriously. And if people appreciate what this type of information, please make sure you're supporting your local independent neuroscientist, biologist, etc. That's what I'd say.
All right, Kevin. Well, thank you so much. It was great having you.
You're welcome.